Hello, my name is Lauren Mellenthin, and I am an REU intern working at the Field Museum this summer. I am working with Dr. Shannon Hackett and her postdoc Chad, looking at the evolution of birds and the traits that make them special. The Field Museum houses one of the largest collections of bird specimens in the world, with over 540,000 specimens. Scientists can ask questions about diversity, behavior, and diet from these specimens that were collected over the last century and a half. This summer, I studied one of the 6 senses, taste. Taste can be influenced by a combination of behavior, diet, genes, and the actual taste receptors themselves. In general, birds do not even possess the sweetness receptor gene, and fish-eating birds have a non-functional umami or meaty flavor receptor gene. Why is this? Is this because they eat fish? What do birds taste? All of these are questions I am looking to answer. Museum specimens make it possible to study tongue morphology for clues about tasting, but it isn't enough to answer questions about behavior and diet. I looked at these questions in kingfishers, which are a diverse group of birds with differences in beak shape, behavior, and diet. This variety makes them an ideal group to look at and investigate differences in fish-eating and non-fish-eating species. My project integrated current science on kingfishers by referring to a recent species-level tree of life, or phylogeny. Kingfishers are appropriate because fish eating has evolved multiple times in the family, so there are scientific replicates of these non-fish eating to fish eating transitions. My project has 2 parts to it. The first part examines the morphology of the tongue in kingfishers. This is tricky because most bird tongues are disposed of in the preparation process. I examined intact tongues from pickled specimens using X-ray computed tomography scans, or CT scans for short. CT scans are mainly used in the medical field to look into the body, but Chad and I utilized the technology for kingfisher specimens. We isolated the tongue bones and reconstructed a 3D image and analyzed it to be able to compare species with different diets. We also used CT scans to visualize what the tongue looks like. I placed digital landmarks on the tongue and generated a 3D image to visualize, measure, and compare tongues. The second part of my project involves molecular sequencing and computational analysis. I use modern sequencing technology to sequence 200 genes associated with sensory abilities in birds, including taste and genes related to the functional morphology of catching fish. We use computers and computer code we've written to find these genes in published bird genomes and digitally extract these genes from those genomes and our own kingfisher genome. These gene alignments were then used to compare across divergent lineages of birds and to create probes, or baits, that scan across species of kingfishers whose tissues we have in our collections. We can identify which species have the genes we are looking for by using our probes, and then we can sequence those genes. The sequences can then help us understand the variation in these genes and their function across species of kingfishers. Ultimately, we want to know if there is a specific genetic pattern associated with how birds see, feed, look, and taste in correlation with their tongues, diet, and behavior. These data and results will help scientists understand how behavior and environment affects the morphology and genetic structure of sensory systems, and how they may have changed over evolutionary time. Thanks to the efforts of NSF and collaborators at the Field Museum, REU students like me will have a chance to contribute scientific research findings and begin to understand the complex world around us.